Hello there, my name is Levi Butler and I am a student here at Boise State University pursuing my undergrad in biological sciences, and I'm going to present a little bit to you on the Notch signaling pathway and how the Notch proteins interact with each other during this pathway. So this is my poster, but for the purposes of this presentation I'm going to take you through a few specific slides so that we can highlight the information we want to look at. So Notch signaling is extremely important in determining cell fate and identity because it acts as a transcription factor. So it plays a role in blood vessel communication and is involved in human vascular function, including blood cell development and artery maintenance. Obviously, these are extremely important because these things can result in things like strokes and heart disease, things that are very relevant to us as humans. 4 Notch proteins are involved, but only Notch 1 has been studied in detail. And as we can see through the signaling mechanism, what happens is this protein ends up getting cleaved off and goes into the nucleus where it interacts with another notch that was cleaved off. We want to know how these specific notches interact with each other because there are 4 different types. In this experiment, we looked at how 4 interacts with all other 4 when they were all in the presence of each other. Our questions are, does Notch 4 have the highest binding affinity, just like we've seen before? And what are the relative binding affinities of all 4 when they're in the presence of all the other 3, as we said before? And at what relative concentrations will we see about the same protein expression when we're doing Western blot? Our first step, as far as running this experiment, is to transfect human embryonic kidney cells with Notch DNA. Now, how we did this was by adding the same amount relative to Notch 4 of Notch 1, 2, and 3, but adding less Notch 4 as we went along, so that we could vary the amount of Notch 4. We've seen that Notch 4 shows more expression, so we want to figure out exactly how much Notch 4 we need to get Notch 3, 2, and 1 to show the same amount of expression. Our next step after we isolated the cells that are transfected was a co-IP, co-immunoprecipitation. This essentially is to just isolate this little guy right here, which is the proteins bound to a little bead. It's how we isolate this proteins from a mess of other proteins inside of a cell, because there's a lot going on. After we do that, what we want to do is run this thing through gel electrophoresis so that we can, in this step, we run it through gel electrophoresis so that we can isolate the proteins and separate them from one another. After that, we can do a Western blot so that we can analyze exactly what proteins are shown in an antibody or two so that we can essentially see exactly what proteins ended up on the membrane. Now, after our experiment, we showed that Notch 1 proteins interact at this site. This is a crystal structure of a Notch complex. We can see that the 2 different notches that are in red interact right here, and this is what we've been looking at in the experiment. Notch 4 we found that has a much higher protein expression and binding affinity at lower concentrations. That can be seen by looking at how we put a 0.7 in relation to 1 to 1 to 1 ratio of Notch 1, 2, and 3 compared to a little bit less of Notch 4, 0.7, or down here 0.2 of Notch 4 compared to the same amounts of 1, 2, and 3. Essentially, we can see that Notch 4 has about the same expression when it is at a 0.2 ratio, which is significantly lower, confirming that Notch 4 is much more expressive. So Notch 4 expression is much higher, as we just said, showing that we got similar results to previous experiments. So further experimentation is definitely needed to confirm precise and accurate results showing these protein expression ratios, but upon confirmation, further research as to how these interactions change the Notch pathway may lead to insight into vascular-related issues such as strokes and heart disease. Thank you.